Assalamu alaikum. So this is lecture number 6 in the Hematopoietic-lymphatic system biochemistry. So in this lecture we're going to talk about synthesis, biosynthesis, and catabolism of heme. Now, so heme is basically a protoporphyrin 9 molecule with an iron atom bound in the middle of the molecule, in the center of the molecule. And heme is a major part of hemoglobin as well as other cytochrome P450 enzymes and enzymes in the electron transport chain. So this porphyrin molecule, so heme belongs to a group of molecules known as porphyrin. Now, these porphyrin molecules overall, they are hydrophobic and they are flat, planar, and they are composed of 4 rings, and these rings are known as pyrrole rings. So this is a pyrrole ring right here. So you have 4 rings, A, B, C, D. That's how they are designated. Now each ring can be conjugated to 2 groups. In case of heme, the groups are methyl, vinyl, as well as 2 groups of propionate. And notice that these propionate groups are charged. And we already talked about heme being conjugated with hemoglobin, saying that the planar molecule, the rings, fit into the hydrophobic pocket of the molecule, and the propionate groups extend to the outside, contacting the hydrophilic amino acids on the surface of the molecule. Now, so let's start talking about synthesis of heme. Now, there are 2 major tissues that are responsible for the majority of heme synthesis, and these are liver and erythrocyte producing cells. Now, in liver, heme is used for production of hemoproteins such as cytochrome P450 enzymes. Now, the rate of heme synthesis is highly variable depending on the presence of certain drugs or as needed. On the other hand, in erythrocyte producing cells, the production of heme is constant. Okay? Now, inside cells, synthesis starts in the mitochondria, then it continues in the cytosol, and it ends in the mitochondria again. So what you have in here is a succinyl coenzyme A, which is a product, an intermediate of the citric acid cycle. It conjugates with glycine into a product known as aminolevulinic acid. This ALA then is transported out of the mitochondria. It gets converted into several intermediates, and then one of them gets back into the mitochondria, and eventually it gets converted to heme. So the very first reaction is catalyzed by an enzyme known as 5' aminolevulinic acid synthase. And there are 2 isoforms of this synthase, ALAS1, which is present in all tissues and liver, and ALAS2, which is predominantly found in erythrocytes. Now, this ALA synthase conjugates, as I said, both glycine and succinyl into the aminolevulinic acid. Now, this reaction is the rate limiting and committed step. So it is highly regulated, and it is a slow as a result of that, and it requires vitamin B6. Now, ALA, as I said, moves out of the mitochondria into the cytosol, where it can be converted into porphobilinogen. So what you will have is that 2 molecules of ALA would attach to each other, conjugate to each other, forming porphobilinogen, and porphobilinogen continues with the other steps that we'll talk about in the next slide. Now, what's important about the enzymes ALA synthase 1 and 2 is that ALA synthase 2 is regulated by iron, so iron dictates the activity of the enzyme in erythrocytes. ALA synthase 1 is regulated by Heme and drugs. So this is liver specific enzyme. So what Heme and drugs do is that they control not only the rate of transcription but also the stability of the messenger RNA. They also inhibit mitochondrial transport. Now, this is the structure of Heme. Now, it differs from the structure of Heme in having iron in the ferric state rather than the ferrous state, and it's also attached or bound with chloride ion. So what you have next is that four molecules of porphobilinogen molecules conjugate with each other, forming a molecule known as uroporphyrinogen 3. Now, this uroporphyrinogen 3 is then converted to a molecule known as coproporphyrinogen 3, which goes back into the mitochondria and it gets converted into protoporphyrinogen 9, then protoporphyrin 9, which then an iron molecule would attach to this molecule, forming heme by an enzyme known as ferrochelatase. So now what's important about there are two reactions that are important. The first one is the one that is catalyzed by ALA synthase, and the other one is the last step, that is catalyzed by ferrochelatase. Now, both of these reactions are regulated, or they are inhibited by lead. So that's why that's one reason why lead is toxic to humans. So again, this is another look for the synthesis of heme. So the first reaction takes place in the mitochondria. ALA is transported out, and it gets converted into all of these intermediates, and then coproporphyrinogen goes back into the mitochondria and it gets converted to heme. Now, defects can occur in any one of these steps in the biosynthesis of heme. That results in the production of a group of disorders known as porphyrias. So porphyrias are basically inherited or acquired disorders caused by deficiency of enzymes responsible for heme biosynthesis. That results in an increase in the level of any one of these intermediates in blood. Now, porphyria, by the way, means purple. So what happens with these individuals is that their lips and teeth and gum would look purple, and skin overall would look purple as a result of that. That also includes urine. Now, these disorders are classified into the affected tissue. So they can be erythroid specific or hepatic specific. And for hepatic dependent or specific disorders, they can also be acute or chronic. Now, these disorders differ in two important things, two important manifestations. One of them is photosensitivity. That is the condition photosensitive or not? Is the patient photosensitive or not? And that depends on the formation of the tetrapyrrole molecule or intermediate. So if you have the formation of a tetrapyrrole molecule having all four rings attached to each other, the patient would be photosensitive, since these ring structures would be able to absorb light. Otherwise, the patient would be photoinsensitive. The other thing is the presence of abdominal or neuropsychiatric signs. There are certain conditions that are related to heme biosynthesis. Now, of course, it's nice if you know these different disorders, but I'll try to focus on two of them. The first one is related to lead poisoning. So what happens? Now, how are porphyrias treated? Well, they are treated by giving patients Heme or hematin. So Heme, as I said, is the heme molecule that has a protoporphyrin molecule that has iron attached to it, but it is in the ferrous state. But Heme has iron in the ferric state, and it is bound to chloride ions. Now hematin, instead of chloride ions, it has a hydroxyl group. Regardless, what Heme or hematin does is that they inhibit the ALA synthase enzyme, the first and most important step in the synthesis of heme. Now, another way of treating porphyrias is by giving patients glucose or prevent them from fasting. The reason is that what happens is that under conditions of fasting, glucagon would be released, and it induces the production of a transcription factor known as PGC1 alpha. Now, this transcription factor is important in the synthesis, in inducing the synthesis of glucogenic genes and enzymes, and PGS and ALA synthase 1 as well. So this is sort of like a result of fasting. By giving patients glucose, you would have the production of insulin, and insulin would reduce the transcription or production of PGC1 alpha, resulting in reduction in the synthesis of ALA synthase and production of heme as a result of that. So let's go into the second half of this lecture and talk about catabolism of heme. Now, there are a number of challenges in here when it comes to catabolism of heme. The first thing is that RBCs are the largest storage place of heme, and they are continuously destroyed by macrophages, now releasing a lot of hemoglobin. Okay, now the thing is that, well, the globin protein is catabolized into amino acids, and these can be used for synthesizing other proteins. But releasing heme is risky because, one, the porphyrin ring is hydrophobic, so heme must be modified chemically in order to make it more hydrophilic so that it can be excreted, or at least it can be handled by the body. Now, the second challenge is that iron must be preserved. Okay, it should not be lost, as I mentioned in the previous lecture. And this is done through a number of reactions. The first reaction is very important. It's catalyzed by the enzyme heme oxygenase. We already talked about heme oxygenase in the previous lecture. Now, heme oxygenase metabolizes heme into a product known as bilirubin. Now, this reaction right here requires NADPH. That's one. Two, iron is released, and I mentioned that in the previous lecture. And then you have the production of carbon monoxide. This is the only reaction in the body where you have production of carbon monoxide. Now, bilirubin then goes through different reactions, and what's important is that the intermediates have different colors. That's why whenever there is a bruise in the skin, you can see that it goes through different colors. First, you have release of blood, hemolysis basically, and then heme is metabolized to different intermediates. Each one of them has a certain color, and that's why the bruise changes colors in here from blue to purple, green, and finally yellow. All of these colors are a result of the different intermediates of heme catabolism. Now what happens is that biliverdin is converted to bilirubin, and bilirubin is then released into the blood, and it is carried by albumin. It binds to albumin, and albumin takes it all the way to the liver. Now, something that you need to pay attention to is that salicylates, salicylic acid, aspirin, as well as sulfonamides, certain drugs, can displace bilirubin from albumin. So bilirubin becomes free of albumin, and it can enter the central nervous system. Now the thing is, in adults it may not matter much, but in infants it can cause neural damage. So you need to pay attention to that. Now in the liver, what happens is that bilirubin is conjugated to glucuronides. So you have the formation of bilirubin diglucuronide. So this molecule is quite hydrophilic, and then it's transported into bile. Now deficiencies in either conjugating bilirubin with diglucuronides or transporting the bilirubin diglucuronide to bile can cause certain conditions, specifically Crigler-Najjar 1 and 2, as well as Gilbert syndrome and Dubin-Johnson syndrome. So what happens, as I said, you have release of bilirubin. Bilirubin is carried by albumin to liver. In the liver it gets conjugated, so you have conjugated bilirubin. Then it goes into the bile, where it can then be dumped into the large intestines. Now, in the large intestines, the glucuronide moieties are removed, producing bilirubin again. Now this bilirubin can then be converted into urobilinogen. So right here you have urobilinogen. Okay? Now urobilinogen, some of it can leak out into the bloodstream. Some of it goes back into the large intestines, and some goes into the blood. Now, the majority of urobilinogen is converted to stercobilin by bacteria. Now this stercobilin is a brown molecule which characterizes feces. Now the urobilinogen that is removed into the blood is taken up by kidneys, and in kidneys it is converted to a final product known as urobilin. Urobilin has a yellow color that characterizes urine. So, knowing the level of bilirubin is very important because it tells us a lot of information about the health of individual, specifically the liver. And this can be done by a specific reaction known as Vandenberg reaction. Well, what's important about this reaction is that it can be it is done in both water and ethanol or methanol. Now the thing is, if it is done in water, then it allows for the measurement of conjugated bilirubin, the hydrophilic bilirubin, and this is known as the direct bilirubin, and it constitutes only 4% of total bilirubin. Now if it is done in ethanol or methanol, these two solvents would allow for bilirubin to be soluble, and it can be measured. So what you have in here is that you're measuring all bilirubin molecules in a sample in order to know what the unconjugated bilirubin, the concentration of the unconjugated bilirubin, then which is known as indirect bilirubin. Simply you follow this formula right here. Total bilirubin measured from the organic solvents minus direct bilirubin, which is measured from the water-based reaction. Since we're talking about catabolism of heme, it's important to discuss jaundice. Jaundice is basically a group of disorders that are related to deficiency in the catabolism, transport, metabolism of bilirubin, and they result in yellowing of the skin in individuals. Now, jaundice can be divided into two classes: either unconjugated hyperbilirubinemia, so there's increase in the amount of unconjugated bilirubin in these individuals, or you can have conjugated hyperbilirubinemia, so it's a condition where you have high level of the conjugated bilirubin. Now, let's look at the different markers related to jaundice in both of these types of hyperbilirubinemia. So unconjugated hyperbilirubinemia can be caused as a result of excessive hemolysis. So you have a lot of lysing of red blood cells releasing a lot of heme. That results in a lot of heme being converted to bilirubin. So you have a lot of conjugation of bilirubin. So you have a lot of conjugated bilirubin in here, going from liver to the intestines, and you have conversion of bilirubin to urobilinogen and sterocobalamin. Now, what characterizes people with hemolytic jaundice is that their stool would be very dark as a result of overproduction of sterocobalamin. But since the glucuronosyl transferase enzyme is overwhelmed with bilirubin, some bilirubin would leak out of the liver into the blood. Okay? Now, so what you will have here with hemolytic jaundice is that there is increased bilirubin in serum and increased unconjugated bilirubin as well. But the amount of conjugated bilirubin would stay normal because little conjugated bilirubin would leak out. Okay? And all of it would go into the intestines. Well, how about urine? Urine would look darker, and the amount of urobilinogen and urobilin would be high in urine as well, since a lot of urobilinogen as well as bilirubin would be taken into kidneys. Well, you can also have what is known as hepatic jaundice. So there is deficiency in the liver itself. There is a problem in the liver itself. So what happens in these conditions is that you would have increased amount or level or concentration of bilirubin in blood because there's leakage. There would also be higher levels of both conjugated and unconjugated bilirubin because again you have cell damage and release of both of these forms of bilirubin into the blood. Since we're talking about hepatic jaundice and we have a problem in the liver, there would be increased liver enzymes as well in the in blood. Urine would look darker because of the release of all of these forms of bilirubin into the blood, which are taken to the kidneys. Now, stool would look either normal or lighter in color, since there would be reduced bilirubin and its conjugates or its forms reaching the intestines. Well, another form of hyperbilirubinemia is known as conjugated hyperbilirubinemia. Now what happens here is that there is production of conjugated bilirubin, but it does not get into bile. or the intestines. So this conjugated bilirubin would leak out of the liver. So there will be increased level of total bilirubin in serum, and there would be higher concentration of conjugated bilirubin since it's stuck in the liver. It has to go somewhere, so it gets leaked out of the cells. Liver enzymes are normal, and urine would look darker, okay, because there would be a lot of, again, bilirubin and conjugated bilirubin taken to the kidneys, which cannot convert them to urobilinogen and urobilin, and that results in reduced amount of these two molecules in the urine, and stool would look clayish as a result of inability of the liver to transport conjugated bilirubin into the intestines, so there would be low concentration of serocobalin in the intestines in feces. Now, jaundice can also affect infants as well. And the thing is, the enzyme glucuronosyl transferase enzyme that conjugates bilirubin with glucuronides is not very active when the enzyme is not active at birth. Now its activity increases with time. So within like 2 weeks, the enzyme or the liver would be able to efficiently conjugate bilirubin with glucuronides. Now, the thing is, the problem is with premature babies. Now, what happens here is that the activity of the enzyme is not really that high, and that results in overproduction of bilirubin that cannot be conjugated, and this bilirubin would leak out. It goes into the central nervous system, and it can damage the brain. So now, usually in normal infants, there would have high concentration of bilirubin, but these high levels are not toxic. But in premature babies, the levels would be very high. They can be very toxic. So a solution is to expose infants to blue light. What blue light does is that it breaks up bilirubin into a more hydrophilic molecule that can be eliminated easily.